Hello, my name is Naomi Pineda, and I am a student at Grand Canyon University. And today I will be presenting about glycolysis, gluconeogenesis, and the role of aldolase gene expression. Okay, so let's just start with a little overview on the main topics that we're going to go over in this presentation. So first I want to give a little introduction of what glycolysis, gluconeogenesis, and aldolase are. So let's start with glycolysis. So it is a metabolic pathway that converts glucose into pyruvate, generating energy in the form of ATP and NADH. It occurs in the cytoplasm of cells, and it is a central process in both aerobic, which is with oxygen, and anaerobic, without oxygen, cellular respiration. Now let's move on to gluconeogenesis, which is the process of glycogen synthesis, where glucose molecules are converted into glycogen for storage in the liver and muscles. Glycogen serves as a readily available source of glucose during times of energy demand. Now, lastly, let's go to aldolase, and it is an enzyme that plays a vital role in various biological processes by catalyzing the cleavage or synthesis of aldol compounds. It is involved in key metabolic pathways such as glycolysis and gluconeogenesis. Aldolase acts as a crucial enzyme in carbohydrate metabolism, facilitating the breakdown and synthesis of sugars for energy production and storage. By facilitating the interconversion of sugar molecules, aldolase contributes to the regulation of energy metabolism in cells. Okay, so now here we have the key steps involved in glycolysis. So the first step is it's glucose phosphorylation. So this is where glucose is phosphorylated using ATP to form glucose 6-phosphate catalyzed by the enzyme hexokinase. Step number 2 would be isomerization. So glucose 6-phosphate is converted to fructose 6-phosphate through a series of rearrangement reactions. Step number 3 would be phosphorylation and cleavage. Fructose 6-phosphate is phosphorylated using ATP to form fructose 1,6-biphosphate, catalyzed by the enzyme phosphofructokinase. Fructose 1,6-biphosphate is then cleaved into two 3-carbon molecules called glyceraldehyde 3-phosphate, also known as G3P, and dihydroxyacetone phosphate, which is also known as DHAP. And our fourth step would be the conversion and ATP production. Here, DHAP is converted to G3P, and two G3P molecules are generated. G3P therefore undergoes a series of reactions, generating ATP and NADH. Ultimately, pyruvate is formed at the end of glycolysis, and the net gain is 2 molecules of ATP and 2 molecules of NADH per molecule of glucose. Now let's move on to aldolase in glycolysis. So hope you remember from the previous slide that this picture is just a little part of the big picture that we saw on the previous one. So over here we can see the enzyme aldolase. So aldolase is an enzyme that catalyzes the reversible cleavage of fructose 1,6-biphosphate, a 6-carbon molecule, into 3-carbon molecules, which are G3P and DHAP. G3P is glyceraldehyde 3-phosphate, and DHAP is dihydroacetone phosphate. If we look back into the glycolysis pathway, we will find the role of aldolase, like I mentioned previously. So the reaction proceeds as follows. First, aldolase binds to fructose 1,6-biphosphate at its active site. Second, aldolase facilitates the cleavage of a carbon-carbon bond within fructose 1,6-biphosphate, resulting in the formation of 2,3-carbon molecules, G3P and DHAP. G3P remains tightly bound to the enzyme while DHAP is released. Step number 3. G3P can continue through the glycolysis pathway where it undergoes further enzymatic reactions to produce ATP and NADH, which are essential for cellular energy production. Fourth and final, DHAP on the other hand, is converted into G3P through the action of the enzyme triose phosphate isomerase. This conversion ensures that both 3 carbon molecules ultimately enter the glycolytic pathway to generate energy. So, in conclusion, aldolase's role in glycolysis is crucial for the efficient breakdown of glucose and the subsequent production of energy in the form of ATP. By catalyzing the cleavage of fructose 1,6-biphosphate, aldolase ensures the proper flow of intermediates through the glycolytic pathway, allowing for the generation of ATP and NADH, which are important for cellular processes and energy production. Now let's move on to gluconeogenesis. Gluconeogenesis is the metabolic pathway by which glucose is synthesized from non-carbohydrate precursors in the body. It is an important process that occurs primarily in the liver and to a lesser extent in the kidneys. Gluconeogenesis allows the body to maintain adequate blood glucose levels, especially during fasting or low carbohydrate conditions when glucose availability from dietary sources is limited. The precursors for gluconeogenesis include pyruvate, lactate, glycerol, and certain amino acids. These precursors undergo a series of enzymatic reactions to form glucose. Here is a simplified overview of the gluconeogenesis pathway. So as we can see on the top, there is glycolysis, and then on the bottom is gluconeogenesis. So it's going to be the same process but reversed. So what we're going to go over right now is the blue section of the image. So first, the conversion of pyruvate to PEP. Pyruvate derived from various sources like amino acids or lactate is transported from the cytoplasm into the mitochondria. In the mitochondria, pyruvate carboxylase converts to pyruvate into oxaloacetate, using ATP and biotin as a cofactor. Oxaloacetate is then converted to PEP by phosphoenolpyruvate carboxykinase, which is also known as PEPCK, and released back into the cytoplasm. Now, let's go over the step of conversion of lactate into pyruvate. Lactate is produced during anaerobic metabolism or in tissues like muscles. It is transported to the liver. In the liver, lactate is converted back to pyruvate by the enzyme lactate dehydrogenase. Next, let's go to the conversion of glycerol to dihydroacetone phosphate, which is also known as DHAP. So glycerol, derived from the breakdown of triglycerides in adipose tissue, enters the liver and is converted to DHAP through a series of reactions involving the enzymes glycerol kinase, glycerol 3-phosphate dehydrogenase, and triose phosphate isomerase. Fourth, the conversion of amino acids to intermediate metabolites. Certain amino acids, such as alanine and glutamine, can be used as precursors for gluconeogenesis. These amino acids are converted to intermediates of the TCA cycle, such as oxaloacetate or alpha ketoglutarate, which can then enter gluconeogenesis. Fifth, we have the formation of glucose. Once the precursors are converted to PEP, further reactions occur to synthesize glucose. The reversible reaction catalyzed by fructose 1,6-biphosphatase splits fructose 1,6-biphosphate into GAP and DHAP. GAP can be converted to glucose through a series of reactions involving glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, and enolase. It's important to note that gluneogenesis, as I mentioned before, requires bypassing the irreversible steps of glycolysis, which is the breakdown of glucose. Key regulatory enzymes such as pyruvate carboxylase, PEPCK, and fructose 1,6-biphosphatase are involved in controlling the flux of the pathway. Hormones such as glucagon and cortisol promote gluconeogenesis, while insulin inhibits it, helping to maintain glucose homeostasis. So remember, homeostasis is when you have a balance. Overall, gluconeogenesis is a complex metabolic pathway that allows the body to generate glucose from non-carbohydrate sources, like I mentioned in the previous slide. It ensures a continuous supply of glucose for vital organs such as the brain and red blood cells, even when dietary carbohydrate intake is insufficient. In gluconeogenesis, the aldolase reaction is pushed in the opposite direction, like I already mentioned. The enzyme fructose 1,6-biphosphatase catalyzes the conversion of fructose 1,6-biphosphate into glucose 6-phosphate, and aldolase catalyzes the subsequent cleavage of fructose 6-phosphate into GAP and DHAP. GAP can then be converted into glucose through a series of enzymatic reactions. By participating in the fructose 1,6-biphosphate aldolase cleavage, aldolase helps generate the necessary intermediates for gluconeogenesis and contributes to the synthesis of glucose from non-carbohydrate precursors. So this enzyme plays a crucial role in the regulation of glucose metabolism and helps ensure a continuous supply of glucose for essential bodily functions. Aldolase, as an enzyme involved in carbohydrate metabolism, exhibits remarkable diversity through its isoforms. These isoforms, including aldolase A, aldolase B, and aldolase C, possess unique characteristics and display specific expression patterns with various tissues. This extensive study of these isoforms has shed light to the diverse roles and highlighted their significance in various physiological processes. The diverse isoforms of aldolase exhibit variations in their tissue distribution, subcellular localization, and functional roles. These differences allow aldolase to perform specialized functions in various tissues and contribute to the overall energy, metabolism, and maintenance of glucose homeostasis in the body. Therefore, understanding the distinct characteristics and expression patterns of aldolase isoforms provide valuable insight into the complex metabolic processes they regulate and their importance in human physiology. So the first isoform would be aldolase A. It is also referred to as a muscle aldolase. It's predominantly found in tissues with high energy demands such as the skeletal and cardiac muscles. Its subcellular localization is predominantly cytoplasmic. Its expression is closely associated with the energy yielding breakdown of glucose in glycolysis, making it vital for energy production in muscle cells. Aldolase A catalyzes the conversion of fructose 1,6 biphosphate into GAP and DHAP, facilitating the generation of ATP and other rich molecules. Now, aldolase B, on the other hand, is primarily expressed in specific organs involved in carbohydrate metabolism. These would be the liver, kidneys, and intestines. The subcellular localization is predominantly cytoplasmic, associated with the inner mitochondrial membrane in the liver. It plays a critical role in both glycolysis and gluconeogenesis, processes responsible for maintaining blood glucose levels, as we have talked in previous slides. Aldolase B ensures the breakdown of glucose in glycolysis and also participates in the synthesis of glucose from non-carbohydrate precursors in gluconeogenesis. Its unique expression pattern and subcellular localization contribute to its function in hepatic carbohydrate metabolism. Lastly, aldolase C is predominantly found in neural tissues, including the brain and spinal cord. Its subcellular localization is found in the cytoplasm and nucleus. Although its precise function is not yet fully understood, it is believed to have essential roles in energy metabolism and glycolytic processes specific to neural tissues. Aldolase C's expression in neurons and its distinct subcellular localization in the cytoplasm and nucleus indicate its involvement in neural energy metabolism and potential contributions to signaling pathways in the central nervous system. Now let's explore a little bit more on the regulation of aldose B expression. Aldose B expression can be regulated by several transcription factors, including HNF1, which is also called hepatocyte nuclear factor 1, HNF3, which is hepatocyte nuclear factor 3, and C/EBP, which is a CCAAT enhancer binding protein, and DBP, which is a D-box binding protein. Let's explore how these transcription factors influence the regulation of aldolase B expression. So let's start with HNF1. It is a transcription factor involved in liver-specific gene expression and plays a critical role in regulating glucose metabolism. It has been shown to directly interact with the promoter region of the ALDOB gene, which encodes aldolase B. HNF1 binding to the ALDOB promoter enhances aldolase B gene transcription and promotes its expression. The binding of HNF1 is essential for maintaining proper glucose homeostasis and hepatic carbohydrate metabolism. Secondly, HNF3, which is the hepatocyte nuclear factor 3, is a family of transcription factors that include HNF1/3 alpha, HNF1/3 beta, and HNF3 gamma. These factors are involved in liver development and the regulation of liver-specific genes. HNF3 beta, in particular, has been implicated in the regulation of aldolase B expression. HNF3 beta binds to specific regulatory regions of the ALDOB gene and activates its transcription. thereby promoting the expression of aldolase B in the liver. Now let's move on to C/EBP, which is CCA8 enhancer binding protein, and it is a transcription factor family involved in various cellular processes, including metabolism and inflammation. It is a member, so C/EBP beta is a member of this family and has been found to regulate aldolase B expression. It binds specific to DNA sequences in the promoter region of the ALDOB gene, and it stimulates its transcription. Activation of CEBP beta enhances aldolase B expression, contributing to hepatic glucose metabolism and glycogen synthesis. So fourth, we have DBP, which is D-box binding protein. It is a transcription factor that belongs to the PAR-B-ZIP, which is proline and acid amino acid rich basic leucine zipper family. It plays a role in the regulation of circadian rhythms and metabolism. Studies have shown that DBP can bind to the promoter region of the ALDOB gene and positively regulate its transcription. DBP promotes aldolase B expression, linking its regulation to the circadian rhythm dependent metabolic processes in the liver. So collectively, these transcription factors, including HNF1, HNF3, C/EBP, and DBP, contribute to the regulation of aldolase B expression. By binding to specific regions of the ALDOB gene, these transcription factors activate its transcription and promote the synthesis of aldolase B. This regulation ensures the proper functioning of aldolase B in hepatic glucose metabolism, glycolysis, and glycogen synthesis, maintaining glucose homeostasis in the liver. In conclusion, this presentation has provided an overview of glycolysis and gluconeogenesis, highlighting the key enzymes, reactions, and pathways involved in these fundamental metabolic processes. We discussed the role of aldolase, a crucial enzyme in both glycolysis and gluconeogenesis, emphasizing its catalytic function and its involvement in specific reactions within these pathways. Furthermore, we explored the different isoforms of aldolase, namely aldolase A, aldolase B, and aldolase C, understanding their structural and functional differences. Each isoform exhibits unique tissue-specific expression patterns, indicating their specialized roles in various physiological processes. The expression patterns of aldolase isoforms were detailed, shedding light on their differential presence in tissues such as muscles, liver, and neural tissues. Understanding these expression patterns enhances our comprehension of the specific functions and contributions of aldolase isoforms in respective tissues. Additionally, we discussed the regulations of aldolase B expression, focusing on the transcription factors in HNF1, HNF3, C/EBP, and DBP. These transcription factors play crucial roles in controlling aldolase B gene expression, ensuring the proper regulation of hepatic glucose metabolism and glycogen synthesis. Overall, the study of glycolysis, gluconeogenesis, and the involvement of aldolase isoforms has deepened our understanding of energy metabolism and glucose homeostasis. This intricate regulation and distinct expression patterns of aldolase isoforms underscore their importance in maintaining metabolic balance and tissue-specific functions. Okay, so I believe we have concluded the presentation, and here is a list of my references. And thank you so much for taking the time to listen to this presentation.